And we'll be opening up to audience questions at the end. So the Slido link that you saw before will be open for the entire presentation, all three presentations, and then we'll be able to roll through all of your questions at the end. We have a pretty long question period that we can hash out anything you might be asking about Verticillium or even other pathologies. So to kick us off, please help me welcome Dr. Harmeet Chawla.
They hear me? Okay, I think they can. All right, thank you everybody. As Jared said, I'm an assistant professor at the Department of Plant Science at the University of Manitoba, where I've recently started working on this disease called Verticillium stripe. Before we get into the disease itself, we'll be talking a little bit about the pathogen and how my connection is with the disease, and then a little bit about the future, how the future looks like for this disease, and how much it's going to be a problem in canola. So beginning here. So verticillium is caused by a disease called Verticillium longisporum. It's a vascular soil-borne disease, and it can pose a significant threat to canola production. It not only infects canola, but can also infect other crucifer diseases, crucifer crops, like cabbages or turnips or anything which belongs to this family. In Canada, this is still a new disease and it's emerging pretty quickly. But in Europe, this was first reported in 1969 in south of Sweden. And from that point on, the disease has really taken off and now can be detected in almost all European countries and almost all cruciferous crop. In Canada, this was found in 2014 right here in Manitoba. In 2014, this prompted a CFIA survey where they could detect it in almost all different provinces now in Alberta, British Columbia, and Saskatchewan. So it seems to be that it's moving westward. So now it's detected very heavily in Saskatchewan, a little bit in Alberta, and also appearing in British Columbia now. Before we get into the disease itself, I want to spend some time talking about the pathogen or the genus which this pathogen belongs to. So Verticillium longisporum belongs to the Verticillium genus, and this genus is infecting a lot of different species across a lot of different crops. For the people or potato growers in the room, they might have heard Verticillium dahliae. So this was a disease which belonged to the same genus, or this was a pathogen which belonged to the same genus. It causes Verticillium wilt, which used to be a pretty big problem in potato. But the Verticillium we are talking about is called Verticillium longisporum. It's a different pathogen, but belongs to the same diseases. But this one has a narrower host range. So Verticillium dahliae could infect a lot of things like potato, tomato, eggplants, whereas longisporum is confined to crucifers mainly. Just to go briefly over the disease cycle. So the disease starts as microsclerotia, which is present in the soil from last year of canola stubble, which is left in the ground. These microsclerotia would start developing as soon as they detect a canola root growing through it in the current season. Once the roots, this microsclerotia starts developing, the fungus quickly moves towards the xylem vessels. So these are, you can imagine them as highways which carry water and nutrition in the plant from the root to the up. So once this fungus has colonized in the xylem, it will block off the nutrition flow to the plant, and hence would cause the disease symptoms which you characteristically see in the field. When we talk about the symptoms in the field, there are different symptoms which you can observe. You can observe long streaks on the stem. This is the reason why the disease is called stem striping and it's not called wilt. So because it's not causing wilting, it's causing these striping kind of symptoms. Gray microsclerotia would also appear on the stem itself. But the critical part here is all these symptoms are appearing fairly late in the season. So if you're scouting at 60 or 70% or 80% seed color change, you would not see them. So you would have to scout very, very later to the season, probably right before swathing. So the reason I got involved into Verticillium was because during my PhD and master's in Justus Liebig University in Germany, Verticillium was a topic of interest, and it was studied very heavily in Europe. Whenever I tell people about Germany, they get all excited and expect that this would have been the case for most of my time. But a lot of time I spent was like this. This is a bar which has converted into a COVID testing center facility. But anyways, most importantly, the asked question from a producer perspective was, does verticillium cause any yield losses? And again, since it has been prevalent in Europe for quite some time, so we look back at the literature from Europe. Mind you, this is winter canola and not the spring canola, which we grow here in Canada. They are testing 4 different varieties of winter canola, and as you can see, this axis is the disease rating. As the disease rating is going up, the yield is dropping. So after, let's say, disease rating of 5, you can expect a yield drop of 80%. But if you look closely to this paper, they are talking about where they're saying about the yield loss. In the very next line, they say that this yield loss was calculated on per plant basis and not on the per plot basis. So simplified, it means that there was yield loss observed at per plant if you're testing them in the greenhouse, but they did not see any yield losses at per plant, per plot basis. Then the question becomes, does it actually cause any yield losses on per plot or here in Canada? We don't have a very, very clear answer to that, but that could be a very, very good question to ask. So this year we partnered up with Manitoba canola growers in their on-farm research program, where they collected soil samples and sent them to us. So here on y-axis you have the yield, on x-axis you have the amount of pathogen which was present in the soil or the stubble sample. And as you can see, as the verticillium DNA or verticillium load in the soil or stubble is increasing, the yield tend to drop. But our challenge is that because of the design of this trial, we can see that the yield is dropping, but we do not know exactly by how much it actually drops to. But there are seed companies which are claiming that they have resistant varieties which can provide decent amount of resistance to verticillium at this point of time. But only time will tell how good the resistance is. But one thing to point out is, from a producer perspective, is that we should not expect to see a blackleg type of resistance where you have a complete resistant or complete susceptibility. It will be more like a disease control. So you'll see a little bit dip in the disease or some dip in the disease, but it should not be expected to have a full control as you have been seeing in the blackleg. Through the Manitoba canola growers on-farm trials, we also looked at other agronomic practices which are often used on the farming on farms here in Manitoba. So we looked at whether straight cut or swathing has an effect on the amount of verticillium or the amount of disease, and we saw that it did not have an effect. This was also so we did this analysis based on a molecular assay. Whereas Manitoba canola growers, they collected visual rating for the disease and pretty much found the same trend to be true. We also looked at early season fungicide, and you can see here that in the untreated sample, there's a little bit of more disease, but it's not statistically significant. So we could say that both of these treatments do not have a real effect on verticillium. Moving on to some of the research areas which we have been exploring at my lab here at University of Manitoba. The biggest question we are trying to answer is if there is any genetic resistance which is already available in the breeding programs. We partner up with the private seed companies and the public breeding program at the University of Manitoba and AFC in Saskatoon. We test those varieties using the methods we have developed and try to see whether there's resistance. And actually there is some good news there, that we have found some very promising sources of resistance, and we are further working on to confirm our results. The second thing we test here is to test new sources of resistance. Particularly, we are excited about checking the progenitors, the species which make up the canola. So we are testing cabbages and turnips. If we can find some resistances over there and bring them to canola. So this is a more long-term goal. Another interesting question we are trying to answer is why certain Verticillium longisporum isolates are more aggressive than others. So as a part of Manitoba Ag's effort, when they go out in the fields and scout them, they would send us samples back and we would isolate the pathogens. What we have been seeing in the lab is some of those isolates have been more aggressive than other isolates. And we are trying to figure that out, why they are more aggressive, or if they are more aggressive isolates. And if there are, where they are in the province. So we can then provide a very applied messaging to the producers of the risk level on their field. Last but not the least, as I touched upon, we also try to study the impact of management practices. This year, going forward, we'll continue some of that work to even further strengthen that work and find some new things. Across the prairies, there has been a lot of effort to actually work work on verticillium and define or identify solutions for the producers. A lot of effort has been concentrated around understanding the genetics and pathology. There's a lot of effort concentrated on identifying new genetic resistance sources and integrating them into the current breeding program, so then that they could be integrated into the variety. There's a lot of people who are collecting different isolates across the prairies, not only here in Manitoba, but also in Saskatchewan and Alberta. They are characterizing them, again, trying to find out whether there are more or less virulent isolates. One of the hot topic is a question which is often asked is blackleg and verticillium interaction. This has also been an active area of research where people are trying to find out whether having blackleg would make verticillium worse or having verticillium would make blackleg worse. So that's another area of research, and it's some interesting pre-results are coming out of that area. The other big challenge with verticillium is if you have ever scouted your field for this disease, it's very, very difficult to actually identify and pinpoint that this is the disease you're looking for. So people, including myself, have been working with improved diagnostic methods, which can be used to identify and characterize the disease. Canola Council of Canada has been heavily involved in standardizing the disease rating scales, because then that scale can be used by industry and agronomists so that we know the exact situation on the ground. Then we also are studying the effect of environment, particularly drought and heat. We know that verticillium is impacted by those two things, and we have been, me and other researchers across the prairies have been studying in that direction. Last but not the least, effect of soil pH. How does this impact verticillium, disease establishment and progression in a plant? And we also have folks at AFC in Morden who are trying to establish field nurseries, so we can get you that answer of how much yield is lost because of verticillium. How does the future look like? Only if I had a crystal ball, I can tell you what's going to happen, but usually there is no research which has been conducted here in Canada on that particular topic. But usually if we do not know the answer to something, we actually turn to Europe. And this was a study done in Europe where they had a facility where they could raise the soil temperature by a couple degrees and see the impact it has on the disease. So what they found was that if you raise the soil temperature by a couple degrees and the air temperature, the disease seems to get worse. And unfortunately, that's the direction which climate change is heading us to, where the air and the soil temperatures are rising. So it is expected that more disease will show up in future, and it will be continuing to show up for the next foreseeable future. So to sum up my talk here, verticillium is now an established disease, not only in Manitoba, but it has been very prevalent in Saskatchewan. The provincial surveys are pointing to this now, that in Manitoba it has been consecutively increasing in the last few years. Saskatchewan surveys have also given the same indication that more and more fields are turning up to be positive now. We do know that yield loss does occur, but it's hard to quantify it at the moment. So we cannot give you a number at this point of time. How much are we looking at? If you are ever scouting for verticillium, the best time to look at would be at swathing, because the symptoms are appearing very, very late in the season. No agronomic practices, at least the one which we have tested right in this study, seem to be working against it. We are also looking at crop rotation, or the length of crop rotations, which might have an effect, but we'll have an answer to that maybe next year. Genetic resistance is the most promising, and we are happy to report that we have found some good sources, and we are working hard to actually bring these sources to the industry and to the breeders so that these can be incorporated into the varieties which you grow on your farms. And of course, the environment matters. Stress conditions, drought, heat, and soil factors are likely to actually increase the disease, and it's going to stay here for a while. So lastly, I would like to thank and acknowledge our funders. None of the work we do can be done without the support from our funders, mainly Government of Saskatchewan, Sask Oilseed, Manitoba Canola Growers, Western Grains Research Foundation, and the SCAP program through the feds. Thank you very much.
A reminder. Hello. Any of your Slido questions will be saved up until the end. So if you have a question for Harmeet, you can scan this here on your phone or ask it directly with the microphone at the very end. Another reminder also, CCA credits are available at the back of the concourse there as you leave. There are CCA credits for John's presentation that came before, this panel of Verticillium speakers, and then also for Brittany Wood's presentation after this panel. That in mind, we can look forward now to Dr. Stephen Fox, presenting from DL Seeds. Give him welcome, everybody.
Oops. Thanks very much for the invitation. I'm really glad to be here today. I've been working with this disease since about 2023, and I've definitely developed the opinion that this disease has teeth, and what that means is eating some of your yield. And that's what I'm going to try and show today. So what I'll be talking about is a little bit about that disease survey that was mentioned, a disease incidence rating scale that I've there, how maturity affects disease ratings, and then talk a little bit about the reliability of doing this. And all of this is so we can speak a little bit about yield estimation, and then a few final comments. So this is the disease survey that's been, it's, I've mentioned 2014 was identified, and you can see that these numbers here have been climbing since they started taking disease survey information in 2018. So now about 3/4 of canola acres has got some verticillium in it. And then the average of that incidence has climbed now to 41. So over here, I've just got what I call a situation triage. So you've heard we don't have pesticides to control this disease. We have host genetics, but we don't know a lot about its genetic control. We do have some genetic tools. We can detect the pathogen in the plant before you can see the disease itself. And we have been taking ratings in disease nurseries, but these have been mostly naturally occurring. As it turns out, we've got lots of places in Manitoba where we can see it now. We also, one of the problems with field nurseries, you only get to look at the disease once a year. And but we've had success in actually setting up the nursery in the field, and we can do reliable disease ratings. We've also been doing some indoor testing, but that is not something that's working very well yet, and we don't get good correlation between what we see indoors and what we see outside. And there are no industry standards, so we're all the different development companies are working on this disease presumably and coming up with strategies on how to manage it. But with time, there'll be some standardization, I'm sure. So I started working on this disease on August 2nd, 2023. I went to a yield test site and I saw this plot here. And I saw it 3 times because it was a replicated test. So I quit what I was doing, started looking at this. This is the same plot again a week later. It's the same hybrid. And this is the same hybrid at a different location 2 weeks later again. So this is bad, and we don't. And then this very last one here, that's the parent of this hybrid the next year. So that's partly why that hybrid is so poor. So I had to come up with a way to rate this disease. So I have a 5-point rating scale. I like to keep it simple. So basically, it's you can't find symptoms, or you can find them, but you have to look in the plot for a little bit. It's not a disease where you can look at a few plants. You have to look at a plot of plants to see it. And we've got These poor ones at the end where there's a heavy infestation, and if it's heavy enough, it'll damage the stems enough so the plot will actually collapse because the stems have failed. And then you've got this intermediate category here, and that's where there's some infection in there, but the majority of the plants are still healthy, or they appear to be healthy. So this slide here is what I would call a poor, it's a 4 up close. And this is sort of the best time, I think, to look at this disease where the leaves have fallen off the plants, but the stems are still healthy, the ones that are not infected, and you can distinguish between healthy and sick plants. And I'm looking at incidence, not trying to figure out if the plant is half infected or a little bit infected. It's just, are they infected or not? Oops, touching something. Yes, so this is the next slide here. And so you have this rating scale, but as I mentioned right at the outset, maturity is affecting the ability to detect the disease. So you have, I'll just come over here. So this is data from 2024. This is 225 hybrids, I think. And you've got these are all the disease ratings from 1 to 5 charted against maturity. And so what you can kind of see here, when you get really late maturity, you got this gentle slope in the data here. And so you don't actually see a lot of susceptibles at this end. And over here, where you've got earlier maturity, you tend to see more. So it means you can see better here, and you can't see very well over here. This thing has pushed ahead all my notes here. So I put these 2 points, I've already mentioned it here. The registration checks that all this data is relative to are the orange dots, and you can see that those are generally not too bad. And but the important or exciting part of this chart here is there are indications of resistant material in the breeding program. So you've got all this stuff here, what I would call very good, that are in all these maturity ranges. So there's some good hope that we can do something with this disease. This here basically just summarizes where those red lines are in that chart in the graph over there. So you have very good at the bottom and very poor at the top. And the trend line that's in the middle separates good from intermediate. And the idea is if you're to the right of that line, it's at a certain rating, it still might be good, but you might be getting fooled as well, which means you have to go back and look again. I got this picture from Justine Cornelison. It's a strip trial that Brett Young grew near Yorkton last summer. And I think it's kind of annoying. What you can see here, well, the yields were quite similar actually, but these are the Verticillium ratings that were taken, and one of them was 4. And you see it because we were starting to see canopy collapse in that state. And interestingly, that's one of ours, they knew in advance that there was going to be problems, and then now you can see it. But it didn't take a yield penalty here. Talk about correlations of ratings. So there's lots of things we're trying to understand here, but correlations between heights have ranged between 0.1 and 0.6. A perfect correlation would be 1. So they're weak to fair ratings. They're not good. And that means you have to look at this disease more than once in more than one place. You'll also see that if you have a lot of disease at a site, you can do a better job. If you have a low amount of disease, you can still detect susceptibles, but you do a very poor job in trying to separate out the very goods and the goods and intermediates. And that's why the correlations start to collapse, because some sites are really good and others are just okay. So this is kind of obvious. Replicated data is really helpful. And a susceptible rating is reliable. A resistant one means you have to go look again. Okay, correlations between raters. There's got to be more than one person looking at this disease. And so this is actually from just near Sun Valley, north of Morden, and it's a heavily infected plot. And these are actually two plots side by side, a more resistant one and quite a susceptible one. And so we had 675 observations were taken in this set of experiments, and it was rated once by one of our staff on August 20th, and I did it again on September 3rd, and I got a correlation of 0.79. And I think that's darn good, given that the person who did the first rating hadn't done that work before. He had a little training, but hadn't done it before, and it's 2 weeks later that I took my rating. So not bad. I think we have some confidence in using that scale. It's something we can use. The last part of reliability is, if I look at the thing last year, and I look at it again this year, am I going to get the same answer? Because that's really important. I only have 36 hybrids here, and I basically compared, got all the ratings I had available where there were 2 years available, and looked at the ratings that were assigned. And so if they were the same, we got a match. If you're 1 step away, similar, 2 steps away, that's dissimilar, and that's not good. The good part of this is 83% of these decisions were good, matched, or similar over 2 years, which kind of means you don't change your opinion of your hybrid. So if it's very good 1 year and good the next year, we're still good. And if it's poor 1 year and very poor the next year, we're still poor. So this is a good BN2. It does mean you have to check again, because there were some that were poor. So I can talk about yield. The problem is, traditional ways of measuring yield loss is, for example, spray, no spray, and to try and tell if there's yield loss. But as I mentioned, we don't have any pesticides to do that job. Another way is to put resistance genes into different backgrounds. So you have the same background with and without a gene, compare those, and see if there's a difference. But we don't know enough about the genetics to do that either. So my approach has been to group hybrids by disease reaction. And then you can compare the disease reaction of these disease reaction groups. We can, and then compare those between different field sites as well, where the disease is present or not. So this describes the data that goes into what I'll be talking about. I've got just this slide, and the fun slide is the next one. But you have, in 2023, for example, we had 224 hybrids, and they were all rated for their reaction to verticillium strike. And then you group all the hybrids based on their common disease. So you had 76 hybrids that were very good, 39 that were very poor, and the others are listed there as well. And a similar structure for the other 2 years. So what you see here is there's a 3-day difference between the early of these groups in 2023. There's only a 2-day difference in 24 and a 1-day difference in 25. Now, if you exploded one of those groups to look at all the individual hybrids in there, that range would be much bigger. It could be 8 days difference between the earliest one and the late one. So the purpose of one of the things these groupings do is it calms down the traits, all the other traits that aren't directly related to disease resistance. And so that, and that why this is important. So what I'm doing here then is I'm assuming that verticillium stripe resistance is genetically unlinked to yield and maturity. And so then if we group the hybrids by their disease reaction, we've minimized anything that's not related to verticillium stripe. So it mitigates confounding effects when you look at an individual hybrid. If it's, for example, high yielding and susceptible versus a low yielding one that's resistant, if you look at them together, they look the same in a verticillium affected site. So you can't figure out what's going on in that situation. Whoops. So what I get out of this, and so the title tells you what I think is going on here. Yield loss estimates of 8 to 18 or 6 to 18% for very susceptible hybrids. That's what I can see. So what we're doing here, you'll first of all look at this this yields here. These are all yields expressed as percent of the registration checks. And so if we first of all look at this one here, the yields look pretty similar at the sites where there was no verticillium. So each of these, and that makes sense because if verticillium is not at this site, then all these disease ratings over here don't matter. It doesn't make any sense. And so they're all pretty similar. A little bit noisier down here, but not to be concerned, I don't think. However, now it doesn't want to work. Okay. If we look at the verticillium affected sites, now you see something that's really interesting. The very good group has the highest yield. And as you become less and less resistant, the yield declines. And this happens in each of these 3 years. So, what we can do now is let's try and isolate the yield loss part. And so, what I've done here is taken the yields of the no verticillium site and used that, and I subtracted these numbers from that to get these ones over here. And that represents the site differences between all the verticillium sites that didn't have verticillium and all the sites that did have verticillium. So there are some differences innately from those sites, separate from being resistant or having disease. So it includes the site differences plus the loss differences. So now to get these numbers here, I'm simply subtracting the very good difference, 2.9, from all of these ones here to get these numbers here. And I'm assuming, and that's because I'm assuming that the losses to disease represent, well, 2.9 represents the difference between the no verticillium sites and verticillium sites. And then when we do this, we're assuming that the very good group is not affected by verticillium, which might not actually be true, but I can't deal with that any differently right now. So once you've done this, so this assumes that there's no impact of verticillium stripe on the very good group. You've made these subtractions. So now we get 7.5% difference, or 7.5% yield loss on a very poor hybrid in 2023. Come on. 6% in 2024 and 18% this past summer. This past summer was the highest level of disease that I've seen in the fields. And if you looked at some individual sites, this number might climb up into the low 20s for yield loss. So just some final comments then. Verticillium is a new disease, relatively, for Western Canada, but we do have experience tools and experience that can be used to put towards this disease. It's not like this is the first disease that's come along, so we have strategies to go and find to deal with it. We're not working alone. We do participate actually with the University of Manitoba. We did share a nursery with Warden Research Center this summer, and we've been sending samples to U of A where they work on this as well. The steering committee, Verticillium Strike Steering Committee, was just started last spring, and looks like the MCGA is going to be picking up the responsibility for operating that. It's an important platform for information sharing. But like other new diseases, just genetic resistance is not going to be the only solution. Besides host resistance, we're going to need some systemic fungicide, agronomic strategies, biocontrol options. With that, I go from there.
Hello? Yes, okay. Another reminder of our Slido questions. If you want to save those to the end, we'll have those and be able to run through. So thank you to Stephen, and I'll hand it over now to Kendall Baker with Manitoba Canola Growers Association. Give her a hand, everyone.
All right. Hello, everybody. So, like the previous two speakers, I'll be taking this time to chat with you about Verticillium stripe, and more specifically, what we as the Manitoba Canola Growers Association are doing to help manage this disease. So to start off, I'm going to highlight our new service that we launched this fall. The Canola Diagnostic Service provides free diagnostics testing to help Manitoba canola growers identify and manage crop diseases. So we offer testing on clubroot, blackleg, and verticillium, as well as a combined blackleg and verticillium test offering. This service is free for our farmer members, and each member can receive one free test type per field. Our website outlines how to submit a sample, and I will have this QR code at the end of this presentation as well, if anyone is looking to learn more about this service. So, as we heard from the previous two presenters, verticillium is a very prevalent disease across our province. And we, as your grower voice, want to make sure that farmers have access to the tools needed to know what diseases are in their fields. Proper identification is our first line of defense. So this graphic outlines just some of the areas of Manitoba where we saw higher amounts of verticillium. Higher amounts of sample submissions come in from. The red colored pins being areas with higher loads of verticillium, and the orange colored pins were still areas with high sample submissions, but had moderate to lower loads of verticillium. Note that any samples that are sent in are going to be a worst case scenario where farmers are already clearly seeing disease symptoms. And this isn't a broad survey by any means, but instead a voluntary service provided for farmers. These pinned areas were just the most common, and there are many RMs that are not represented on this map that still had verticillium loads present in 2025. Moving on to some ongoing verticillium research. Since we know that verticillium is a major problem for farmers in Manitoba, we at MCGA put 43% of our total research and agronomy budget toward research project funding this past year. A good majority of these dollars go towards verticillium research projects at Canadian universities and Ag Canada. As we know, there's still a lot of discovery and groundwork that needs to be done to understand verticillium better. These are the verticillium-focused projects we're currently funding. Using CARP 2024-43 on the bottom there as an example, for every $1 MCGA supports, our co-funders provide $5. This means that canola growers' dollars are extended, and in return, impactful research is provided back to farmers. And as results from these projects become available, we will be showcasing them for farmers. Websites like the Canola Research Hub are a great way to easily access Canadian canola research projects. Our canola counterparts in Alberta and Saskatchewan showcase the projects they fund on here as well. The hub can be found on our website under the Research tab. After the 2024 field season, our board wanted us to focus heavily on some more applied Verticillium research project or research solutions in our on-farm research program. So in came the Applied Verticillium Research Initiative project led by Dr. Harmeet Chawla, focusing on these 4 objectives. So 1, investigate harvest management practices, swath versus straight cut, effect on verticillium incidence and severity in current year, and inoculum return to the soil. 2, evaluate early season fungicide applications and how co-infection with blackleg and sclerotinia may influence losses. 3, a potential diagnostic soil test for microsclerotia per gram soil for farmers to understand risk. And 4, better understanding of farm management practices related to verticillium incidence and severity. Now, the question stands, how do we take all of this important verticillium research and make it useful for farmers? By conducting trials directly on farms across Manitoba, we can evaluate new and improved production practices under real-world conditions that reflect the diversity of soils, climates, and farm operations that growers actually see day to day. This on-farm research approach allows us to answer applied research questions that can't always be fully addressed through small plot or greenhouse studies, while also better understanding the likelihood of success and economic impact at the field level. We at MCGA are committed to bringing research closer to the farm and putting farmers at the center of the process. By working alongside growers, research partners, and industry, we ensure results are useful in the current climate and are transferable to the field level. We are working to bridge the gap between traditional research and on-farm adoption, so farmers can confidently use our research findings to make informed decisions that improve productivity and profitability on their own operations. In 2025, our research team brought in 2 new on-farm research trial protocols to support the first 2 objectives from the Applied Verticillium Research Initiative project I referenced previously. They were called Harvest Management and Early Season Fungicide. The Harvest Management trial compares swathing and straight cut strips while using overlaid survey style data collection to understand if we can see any trends with historic management in canola fields and their verticillium pressure, while additionally understanding spring soil sample levels of verticillium and what that means for fall pressure to see if we could potentially use it as a decision tool for directing farmers to what fields they should grow canola in. After we collected all of the 2025 harvest management trial data, the main takeaways were that the swath versus straight cut plots had no significant effect on yield when looking at combined data across all trials. The main takeaway for verticillium was the same story. No significant difference in verticillium incidence or severity when comparing swath and straight cut plots at 80% seed color change and post-harvest. These results are to be expected as there was really no actual treatments that were applied other than operation timing. The interest lies with the 2026 spring soil sample. For the early season fungicide trial, We have participating growers tank mix essentially a blackleg fungicide at herbicide timing. All treatments are replicated 4 times, and positions of the treatments are randomized with each replication as well. Treatments used were Nexicor and Maxentis. We are looking to see if this early timed fungicide application aids in the crop's ability to resist losses from Verticillium and how co-infection with blackleg and sclerotinia may influence this. In the 2025 season, we saw very low incidence of blackleg, including 2 trials with 0 blackleg. Considering the goal of this trial, the use of blackleg fungicide in the absence of its target pathogen should have no effect on Verticillium incidence or severity. With that in mind, we can't say that blackleg fungicide has no effect when Verticillium and blackleg are present at the same time, because we didn't see serious examples of blackleg infection. The important takeaway is that blackleg fungicide does seem to obviously reduce blackleg when it is present in low amounts, and the suppression of blackleg inoculum for future seasons may provide indirect long-term benefits. Again, these 2 trials were new for us in 2025, and we will be continuing them in partnership with Dr. Chawla's lab at the University of Manitoba for the 2026 season. In addition to diagnostics testing, funding Verticillium research projects, and running our own Verticillium-focused on-farm research trials, we also sit on a Verticillium steering committee made up of industry experts. Through this committee, we provide a grower voice and help ensure that farmers are actively involved in the decision-making process, alongside seed companies, researchers, and universities. This collaborative table brings together all parts of the industry to address Verticillium in a coordinated way, and our involvement helps keep grower needs and priorities central to the conversation as solutions are developed. All of that being said, and even though we don't know the answer to this problem, we are trying to understand and look at some management solutions that farmers might be able to implement in the shorter term while we wait for longer term research projects to come up with the answers that we need. So please reach out to us if you have any interest in being a part of the harvest management or early season fungicide trials or any of our other on-farm trials. My contact information is here. You can reach out to me. It's also in our 2025 on-farm research booklet that we are handing out at the top. And you can come see us at our booth just down the main concourse as well. Thank you.
There you go. All right, thank you to our full Verticillium panel. That closes it for the actual presentations, but I encourage you to stick around. Actually, we have a whole list of questions I was just checking out while we were seeing the end of the presentation there. So some really interesting questions, on-farm decision quality information that is being asked, of course, by the farmers. So I ask our panels now, we can move the chairs over and start the actual panel discussion here. So over to the front of the screen. Down here. Okay. So we actually had to pass the mic over for Harmeet. So if you want to answer, I can hand it over. Oh. Okay. So I will start us off with the first question that I see here. How long is the pathogen dormant in the soil? Verticillium specifically.
We don't know yet, but what we know from the European studies is that it can stay in the soil for up to 6 to 8 years. We know that Verticillium dahlia can stay for more than a decade, so pretty long. So the crop rotation might not be very helpful, but it might decrease the fungal load over time.
Okay, and this question actually relates to something I talked about with you, Stephen, earlier. Does seeding early versus late May potentially make a difference in verticillium severity? Because I remember you talking about earlier or later
I
senescence.
don't know, but that's something we should be looking into. It's a lot of questions like that that deserve an answer.
And so would you have a plot design or experiment design in mind if you were to start testing that? And is that something that you would do with DL, or would you like to collaborate maybe with AFC or U of M and looking towards that?
Well, they're agronomic questions, and plant breeding programs are not really very good at dealing with agronomic questions. I'll do that.
Maybe
But we've got lots of things we could try, but we need a place to do it.
maybe jabbing a little elbow at MCGA saying, hey, maybe that should be an on-farm trial that we ourselves are doing. Okay. So one maybe broader question here is this genetic solutions related. Is this something that all breeders will have access to, or is it company specific? I think he might have been talking about the resources available at DL Seeds that you're seeing now.
Well, every company has their own genetic resources, and I'll certainly explore those. But we're always interested in other sources of resistance, especially if you've got some sort of simpler genetics to offer. That's of great interest for plant breeding.
Okay, thank you. One environmental question. What is your hypothesis on the effects of drought and temperature on canola with verticillium spread?
Can't say about the temperature part because we haven't been checking that for now. But for the drought part, it looks like drought, drought actually makes the plant more resistant to verticillium. So a dry spring would help for not having the disease. What we have seen is that once you expose the plant to drought, it naturally forms some barriers in the cell to protect itself from drought. And those barriers are then actually could be useful to prevent verticillium from infecting. So drought would be good in terms of verticillium, but the drought itself would cause some yield losses. So, but we haven't checked for the temperature part yet.
Okay. So this one's for you, Dr. Fox. You said that this may have been the worst year you'd seen for verticillium. Was it, would you say, environmental conditions or just the progression of the disease across Manitoba?
I like option 2. You can see it but we had really good conditions this year. But I'm not sure what those conditions were that were so special. But certainly, the level of infection was eye-opening. Something.
I'm thinking actually about Sakaria's genomic selection models that he's utilizing, where he's incorporating many different environmental factors into the yield of canola in your program. Do you think that could be incorporated to maybe evaluate or predict what verticillium severity could be?
Maybe.
If you have a base.
The thing is, you need the data for it. We don't have lots of data yet. So we're doing that. Acquiring that information and see where it takes us.
Many of these tools that are in hand, that we're at such an early point with verticillium that setting that flag in the sand right now could be a good time to actually utilize these new technologies. So you were talking about your own rating system at DL. Are there different ratings that maybe other companies are using, and are you using some sort of communication to align those ratings between maybe U of M or AFC or other breeding companies?
I'm aware that other people are doing other things, but I think early on it's okay to do it the way that you feel comfortable with. And like what I showed you here, it seemed to work. Like I've said to people, my colleagues, you can't make up this stuff. So you you rate the disease, and then you see what happens. Now, we could get more granular than 1 to 5. And so I suspect we'll end up with a 1 to 9 scale. I'm not that interested in a scale that tries to take apart how much of the plant is infected. I'm using disease incidence, and that seems to be fine.
I think there has been some effort through Canola Council that they have been working on a disease scale, and through the Verticillium steering committee, they have been trying to bring everybody on the same page with the scale, and I think it's now very finalized. And the hope is that the industry would take it, or most people would use it, but I can't say how many of them are using this very scale at the moment.
Okay, so I'm going to be greedy for a second. I actually have a question that I kind of wrote down yesterday after our conversation. So Stephen, Jay, and I talked a bit about the responsibilities of plant breeders across industry. So industry, academia, and publicly funded research, including AFC. An example we talked about was Sally Vale's populations in Saskatchewan, and how resources like that then become available to seed companies and other institutions. So some might think, why don't companies like BASF or DL Seeds just make those populations themselves? And why are we kind of doubling up on efforts from AFC? And it is true for some populations, but could you guys maybe paint us a better picture as to the resources available to each of these institutions, each of these sectors, and the strengths that each brings to the table to solve the issue that we're talking about today, or issues in the past like blackleg or clubber, whatever it might be? It's a broad question, but it's something that I see being important to what every seed that gets seeded is.
Well, yet you're right by saying that each organization does bring different tools and strengths to the table. Breeding programs have a lot of infrastructure that we can utilize. Having multiple yield test sites across Western Canada, that's a very powerful tool at our disposal, which a lot of academic players don't have access to that. But I think, for example, in academic situations, certainly Agriculture Canada is well finding new genetics. That's a really useful activity to be doing. And any new genetics that come out there, we're going to be interested in trying.
I think I can add to that, particularly to Sally's or other people who work at the same center. Usually these guys are also looking at wild relatives of the canola. So sometimes what you can, if you're lucky, you might end up in a qualitative type of resistance. But they have very interesting material which they produce for different projects, not only for Verticillium. So that's their strength, which they bring to the table, and we usually do screen those materials. It's a long shot, but maybe there is something to be found in those germplasm.
Okay, thank you. I have another one here, actually. You touched on it, Harmeet, about the interaction between Verticillium and blackleg. There's some discussion that they may cohabitate on canola. They may be competitors in canola. Would you be able to share a pathologist insider view into what you're seeing maybe in your own populations, your own field trials, and what you're hearing amongst other researchers? Because a lot of farmers around are seeing that, hearing that, but the actual clear-cut answers aren't quite there. And they may not be here right now, but at least if you give an insider view on what you're seeing between blackleg and Verticillium.
The popular opinion is, again, it's a popular opinion, not backed by science, is that blackleg infection would make Verticillium worse. But that's what people have been saying. We don't have blackleg on our field, so it's hard for me to comment on that. Both are fungal pathogens, both infect through the roots, so they are competing at the same place. but it's hard to say at the moment what the interaction is, and probably has to be some more studies which have to go into that direction. Stephen, you want to add something?
I'd add to that, but it's kind of a simplistic view. Farmers really aren't interested in having resistance to one disease and not the other. They probably want both. So that's the goal, is to have both. And if there's an interaction, well, that's nice, but it doesn't really matter because you're going to have both types of resistance anyway. So.
Okay, thank you. So, to your knowledge, what fungicides are potentially on the horizon to manage Verticillium stripe? Have you seen any chemicals or any other applications that are potentially seeing some effects? I have heard of Saltro being used in some strip trials, but that is not an official treatment for Verticillium in any respect, and it also wasn't really quite a replicated trial either. But anything on your end that you guys have seen?
No, I'd like to think that the people in that business are looking at it, because it seems like something obvious to do, but I'm not aware of anything myself.
Yep, totally on board with Stephen on that. And I think there's also people looking at RNAi as an option for fungicide, as a fungicide. So that's, I think, some things getting done in that direction as well, which might come up as an interesting option at some point of time.
For sure. And its nature as a soil-borne pathogen obviously does make that more difficult to deal with. I mean, our discussion, Harmeet, earlier about setting up yield trials, one of the hardest parts about detecting and calculating the amount of yield that you lose through Verticillium is that you can't really, it's very hard to set up a negative Verticillium strip. Can't really spray for it in a strip, and it's very hard to actually detect where the patches are accurately. So yeah, the actual application of fungicides for that also becomes an issue, or fumigation. Straightforward, or actually maybe not straightforward at all. Would tillage make a difference in Verticillium prevalence or timing in the soil?
Don't know, maybe. It could just spread it around more too. I don't know.
Yeah, that would have been my guess as well. Like, I think it'll probably make it worse because you're kind of spreading it evenly in your field then. But I don't have an answer. I don't think anybody has looked at that.
To anyone that has suffered from clubroot and heard the idea of combining that with any kind of tillage, you definitely know the red flags not to do that. If you've ever seen a clubroot map, they can be very beautiful straight lines that follow exactly where your combine or your tiller runs along. Okay, I think I have one, maybe two more questions before we switch over to our last presenter. Okay, so there are a lot of phenotyping and genetic marker detection and introgression tools available to geneticists and plant breeders that we didn't have at a time when we were first struggling with blackleg or clubroot, for example. So what do you see being the most useful tools or techniques that are available at UFM or DL or any other plant breeder finding an answer to Verticillium stripe? And are there any other traits or diseases that you're working on that you're excited to use these techniques on?
Well, I think for most development companies now, we have lots of genetic maps of canola, lots of them. And so we've got this whole set of maps available to us, and all it needs is phenotypic data to be fed into it to try and establish what kind of genetics we're dealing with. And so, those kind of facilities you didn't have really 10, 20 years ago, and now you do. And so it makes tackling this problem a lot easier. And you can get to start trying to at least develop some useful questions to try and answer. And beforehand, we didn't have some of that facility. So I'm positive that we can do something.
Yeah, I totally with Stephen on that. Like genetic maps, more reference genomes are available. There are more molecular marker platforms which are available nowadays. those all will help. And I think another thing is that Europe has done quite some research into this area. They have some things which they have learned, some of which would be applicable here. So it's not a totally new disease. We're not reinventing the wheel. So yeah, I think we will have a solution at some point of time with those resources in place.
Okay, thank you. So I've got one quick one and then one last one. Okay. There was one question about, has there been any work done on spore movement yet, ranges, etc.? So that could actually be potentially for you, Kendall. A quick look at the harvest management program, harvest management trials.
Looking at spore movement, you said?
Spore movement or spore prevalence over time.
Yeah, so I mean, speaking on behalf of the harvest management trial is really the only one that we have kind of conducted through that. And that's the one that's looking at swath versus straight cut strips. You might have more on that one, maybe, but we did look at that a little bit. So
that is early stage right now. We've taken our first field season of looking at harvest and straight cut. Straight up yield, there was no real significant difference in the 2025 season, but the real interest of those trials comes in 2026 when we actually look at the exact same plot areas and see whether or not there's more or less verticillium spores being returned to the soil, depending on if you're chopping it early with a swath or if you're turning it all to chaff at the same time with your combine. Okay, so we've got 5 minutes before Brittany's presentation, and I've got one question that is related to the $1 million question of yield loss, yield curve, whatever it might be. So in the same area as that, farmers want to know what level of verticillium infection results in what yield loss. As you said, the exact relationship isn't a clear calculation yet, but we at least have diagnostic labs already working on tracking verticillium across the prairies, but can be reporting verticillium severity in different ways. For example, DNA of verticillium in the soil, DNA in a tissue. We could do DNA per gram or copies of DNA per total DNA. But could you explain, Harmeet, we had a discussion about this, how you interpret these metrics and what you could see being the most reliable metric going forward for yield loss in the future?
I also would like to have an answer to that question, that how much DNA in, or how much verticillium in whatever form, either is its disease rating or gram of DNA per gram of soil. I think that the challenges, like especially in Manitoba, as you explained earlier, we do not have a site which is not verticillium infested. Most of them are. All the matrices give some useful information. I think it depends on the context you're trying to understand. If you are talking about the disease loss, I think the DNA measurement could be more quantitative and reproducible rather than disease ratings, especially with people using different scales. I think the challenge, again, another challenge is do we, like, for example, I would love to have a number of the most popularly grown varieties, how much yield loss they suffer. But usually you get into that tricky area where seed companies don't like us fiddling around with their varieties and trying to find that out.
Do you have a thought on that, Stephen, on maybe the most reliable metric going forward?
One of the things I'd like to understand much more is it's about when the pathogen gets into the plant. So the susceptible ones, as I mentioned earlier, we have DNA tools that, and you're using as well, that you can detect the pathogen in the plant even before you see the disease. But if you have a really susceptible line, you can detect it sooner than a more resistant. So what does that mean? Like, how do you take advantage of that or understand it? And if it's this much later, then you're okay. If it's really early, well, you're not okay. So I think using DNA tools that way might be useful to try and understand how the pathogen actually progresses through the plant. You know it infects early after seeding, but you have to wait to the end of the summer to find out what happens.
Okay, so Verticillium, clearly top of mind for our researchers and our farmers. Undeniably a focus. We'll be continuing on our research and our outreach with MCGA. I'd like to take a moment now to thank our Verticillium panel, put our hands together for the pros.